All right. So with that, welcome everybody. Thank you so much for coming. This is the first event of the year, so we'll start with a small introduction on what the event's about. So Chilloquium is a weekly speaker series that's aimed at physics undergrads, but is accessible to a broader audience. And thank you so much everyone for coming. And to quote Nirvana, bring a friend next time. So it's a weekly speaker series, and there are 16 of these talks. So the topics will range. We chose them to be as broad as possible, so you'll get some sort of exposure to not only today's talk on information, statistical physics, and biological systems, but also on the astrophysics side, on how the first image of a black hole was captured, and on the more quantum information side, on how to build a quantum computer, among other things. So it's a really wide-ranging thing, and we want undergrads to be exposed to as many things as possible. So it should be a very exciting semester. So we have 16 talks weekly, typically Tuesdays at 4:30 p.m., but we'll send announcements for some special cases. There are a few brave souls from Europe joining us. Not everyone's like Professor Louis in that he can meet at 9:30 over there. Some of them have opted for earlier hours, so we'll make arrangements accordingly. Chris?
Cool, thank you. So just a quick introduction to me. I'm Chris. I am the other co-president of Harvard SPS. And I just wanted to let you in on sort of the philosophy of Chilloquium. So we wanted to kind of address some problems that we noticed in like typical physics talks. One problem is that they're often inaccessible to almost everyone, unless you're in that field or a postdoc or sometimes a professor. So for Chilloquium, we want it to be accessible with a little bit of undergraduate knowledge. And maybe you won't get everything out of everyone, but we hope that it'll be a much more engaging talk than a lot of talks that might be offered that are aimed at more senior students. And then the other issue we wanted to address is the talks always talk about the speakers as scientists. We also want to learn a little bit about our speakers. So typically the speaker will give some sort of an introduction during their talk where they kind of talk about their journey and how they got to where they are. And these are usually really interesting, and they're one of our favorite features of Chilloquium. So just to give you a little bit of an idea of the format, it's somewhere between a lecture and a seminar. We really encourage questions, but there's also a lot that each speaker prepares and is ready to talk about. And the way we're going to try and handle questions is we'll ask that you send them to either send Buddha or I, and we will just check them to make sure they're good to go, and then we'll let you know, and we'll sort of call on you. We like people to ask questions directly because we want this to feel kind of conversational. If for whatever reason you don't want to or you can't, feel free to ask us. We'll ask the question for you. But yeah, if you have any questions at any time, just if you can type them out and chat them to us. And without further ado, I'll go ahead and introduce our speaker. Last semester, we did very biographical introductions, and we thought that was very boring. And you've also already read it and are going to hear it again. So we thought we'd go with fun facts this semester. And our fun fact about Professor Louis is that he grew up with a chimpanzee. And to find out what this means, just stick around. I think there might be some pictures involved. But with that, I'll pass it over to Professor Louis.
All right, so thank you very much to you guys for the introduction. It's a great pleasure and honor for me to speak to you. And I'm going to try to share my screen, just see if does this work. Great, so I'm going to talk about something which I hope you'll be understand with basic undergraduate background knowledge. And stop me at any point if there's a question. I'm very happy to take questions. I'm going to ask a big question really today, why the world is simple. But before I do that, I want to kind of get into some autobiographical information. So here's a picture of myself when I was a child in Gabon in Central Africa, where I grew up with my chimpanzee, Bertje. So my parents worked for a small mission school in the jungle of Gabon. Gabon is equatorial Africa. It's one of the world's great rainforests. It's a country with one of the largest fractions of its land being national parks. And my parents are both biologists. And so we lived in a very remote area. The school catered to children who lived, or actually there were high school kids who lived in villages along the river. And so they came to the school and lived there, like a little dorm school in the middle of nowhere. You had to drive half a day to the nearest town that you could buy anything reasonable, for example. And so hunters, so the only meat we got was hunted meat, and hunters unfortunately shot Bertje's mother without realizing that Bertje was there as a few weeks old. And so we took him and raised him, fed him milk in a bottle, etc. And so he was like a little brother to me. And so I grew up, walking around barefoot, living in the jungle for a very large fraction of my life. When I was 17, I moved back to the Netherlands, which is the country of my nationality, because I wanted to study physics, which wasn't available in Gabon at the time. And it was an enormous transition for me. I had never used a train or a telephone, or barely used a telephone. This is in the pre-mobile phone days. That dates me a little bit. And I hadn't watched television. I didn't really know my way around at all. But I spoke Dutch because we spoke Dutch at home. In Gabon, people speak French and a number of other languages. And so I had a very kind of unusual experience. This is because I looked Dutch and sound Dutch, but wasn't, and so clearly behaved in odd ways. I was a kind of hidden immigrant. And I would sometimes purposely when I realized I'd made a faux pas of some kind, because people were looking at me with odd eyes, like I'd done something wrong. I, of course, didn't know what I'd done wrong. So I would put an error into my Dutch, and people would say, "Oh, where are you from?" And they would be kind of relieved that I wasn't entirely crazy, but perhaps from somewhere else. And I say that because I think it's interesting how many scientists I've met who have been outsiders at some point or the other. I think science is one of the great things about science is that it actually values outsiders. Something about being an outsider, from being different from everybody else, being a minority in one way, that actually helps you to think differently about things. And it's something that we really value in science. One of the things that I love science is part of a culture of liking or having space for people that are different in all kinds of ways. People who are maybe, socially different, mentally different. We all kind of are united together in this idea of trying to uncover something new about the world. And I've loved science from as young as I can remember. I walked, as a child, I walked around in Gabon, in the jungle where we lived. I once counted, I think, 25 different types of fruit and leaves and flowers that you could eat in the jungle where we lived. And I often came to dinner not being hungry because I'd eaten something that I'd found. And I was just remember walking around in the forest and just feeling the majesty of it and the glory of it, and wanting to understand, how does this whole thing work? I went to my undergraduate in Utrecht in the Netherlands, which is a great place for theoretical physics, and I absolutely loved being there. And it kind of, even though I was probably behind a lot of my Dutch peers, because my education had been a bit patchy, I used that time to try to catch up and kind of get back to kind of get up to speed. And the Dutch system is relatively flexible, and it allows you to kind of do that, which was great for me. And I just loved theoretical physics, absolutely adored it. And I went to the US for my PhD. I did my PhD at Cornell University in kind of basic quantum condensed matter theory with a guy called Neil Ashcroft. If you do solid state physics, you'll use Ashcroft and Morin's textbook. So he's a very great person to work with. And I really enjoyed that. And he's very rigorous and very quite formal. You had to, in order to meet him, you had to make an appointment with his secretary, and you come in, and I was always slightly nervous. And I would tell him whatever I had working on, and he'd stop, and he'd always says, but what's the physics here? And it made me stop what I'd been doing, which is typically some kind of long mathematical derivation, and think about, what does it all actually mean? And he was a very hands-off kind of supervisor. He kind of let you do your own thing. And in retrospect, later I realized often he actually knew better than I did often what I ought to be doing, but he didn't tell me. He wanted me to discover on my own, which is a great way to learn, but was also somewhat, difficult at various times, especially when I was completely lost. I remember spending an entire year doing a certain calculation, and at the end of a year working on it, realizing that it was a complete dead end. And Professor Ashcroft would never said that to me, that it was dead end. He'd just say, well, let's think about the physics. Have you tried this? Have you tried that? And he would make various kind of statements that seemed very Delphic to me. And it was only a few years later as a postdoc that I suddenly solved the problem that he'd actually originally given me. And at that point, I realized that a bunch of his Delphic comments were exactly what I had worked out, and that he somehow knew the right way ahead, but didn't want to tell me, wanted to let me kind of figure it out on my own. And so I think probably let me flounder for a year because he thought that was good for me. And it was, I learned a lot from that. One of the things you'll realize when you move into research is that you can have long fallow periods, dry periods where nothing really works, and you can feel terrible, and you can feel terrible about yourself, particularly if you're not careful and allow your sense of self to be linked to how your work is going. One of the great things about being a PhD supervisor now is that I've got a bunch of students, and there's always at least one who's doing something exciting, so I'm always happy. But when I was a member that year in Cornell, just working on something and just grinding, and it never went anywhere. And it took me, after a year, I gave up and went in a different direction, which ended up being fruitful. So that's what I did, a PhD. Then I got interested in more statistical physics. I actually, there was a French, or actually a Luxembourgish French scientist, Jean-Pierre Hansen, that I really admired. I wrote to him, and he in fact remembered me from my undergraduate days. So when I was undergraduate, I'd solved a problem which is relatively well known. And so I wrote to him, and he wrote back to me the same day saying, fine, I'll offer you a postdoc. Never asked me for a reference letter or for a CV or anything. He just remembered me from having met me when I was an undergraduate. So it might kind of made me feel that my whole PhD would have been for nothing, because he gave it to me based on my undergraduate work. But he was moving to Cambridge at the time, so I moved into Cambridge. He actually moved from physics to chemistry. So I ended up in a chemistry department in theoretical chemistry in Cambridge for a number of years. And I really enjoyed that. I was working on a bunch of different things. And one of the things you'll probably see if you move on in academia is, there's actually quite a short pyramid. So there's lots of undergraduates, and there's a small number of PhD students, there's a small number of postdocs, and there's a very small number of faculty positions. So by the time you get to that postdoc level after your PhD, it gets extremely competitive, and can be quite stressful, actually, because you're trying to make your way in the world as a way, and to try to mark yourself out as somebody important. You want to do something important that's much harder than it seems, right? So I will tell my students, when you're an undergraduate, it's really all about answering questions that other people have posed that have answers. And so you're given a bunch of work to do to learn, and then on the exam we give you some permutation of this, but there's always an answer. And so it's all about being good at answering questions. But when you're doing research, it's very different. It's not about answering questions, although obviously you need to answer questions. It's about asking the right question. There are many really interesting questions that are unanswerable, like, what is a fundamental theory that combines quantum mechanics and gravity? Very well-defined question, at the moment unanswerable. There are many questions that are perfectly answerable, but are totally uninteresting. Take calculation from Professor X and tweak it to one more order in an expansion. That's perfectly doable, but uninteresting. The key is to try to find the question that is both interesting and solvable. And that's a very different kind of set of skills than just answering questions like you do as an undergraduate. It's much more nonlinear, and it's much harder to predict when things are going to work well or not work well. And so the other thing about being a postdoc, you're trying to kind of make a name for yourself, and there's lots of politics around this, about going to conferences, giving talks, getting credit for certain things, or seeming to be working on the right topics and the cool things. And I remember I had a colleague who was a very distinguished scientist. It was already then he was quite distinguished, now he's become very distinguished, who really stressed me out, because he always talked about, what is in favor at the moment, what is getting published in the right journal. So physical review letters is a typical high-end physics journal. So what's getting in physical review letters, that's what we should work on, that kind of stuff. And I was getting more and more kind of into that kind of frame of reference, as trying to work on things that I thought would get into physical review letters, things that would get published in the right journals, etc., etc. And one day I was praying, actually. So I'm a Christian, so I pray, and I suddenly thought to myself, one day I'll stand before God and I'll give an account for how I use the talents that I have. And I thought to myself, if I say to God, I worked on the cool problems that I thought were going to be published in physical review letters, it just didn't sound like it would really cut the mustard. I thought I should probably work on something that's important. And it was a really kind of big thing for me. So I took a step back and I thought, okay, I'm going to stop what I'm doing and think about what do I think is a really important problem. And I'm going to try to work in some way on that important problem. I knew at that point that just saying that, I'm going to work on an important problem like unifying quantum mechanics, gravity, that's a fool's errand. You've got to look at who you are, what your background is, what your skills are, what your kind of particular things you can do well, and try to find where you can make the best contribution. But you shouldn't try to look at what's fashionable or what's getting published at the time, and try to find something that's important. Now, I've got a lot of colleagues who are not religious at all, who seem to make this step without any problem. They do this naturally. But I needed that kind of, divine perspective, if you want to think about it that way, to force myself to think, gosh, I've gone on the wrong track. I'm obsessed by what's going to get published in the right journals, and that I shouldn't do that. And there's a kind of tyranny of this in academia, where you can it becomes like a career where you're trying to do what's going to be right to get your career ahead, rather than thinking about what's really important. And so I took a step back and I thought, okay, what's really important? And I realized one of the things I've been fascinated from since a child, since having Bertje actually, is what makes us different. Like, what's different between myself and Bertje? It turns out that we share a lot of DNA. In fact, we share something like 98% of our DNA with chimpanzees, depending a bit on how you count. And although we look differently when we're young, my mother said we behave remarkably similarly. But over time, we diverged in a lot of important ways. And in fact, it turns out that we share huge amounts of our DNA with other animals. We share 50% of our genes with E. coli bacteria, 25% with yeast, 70% with frogs. Even, if you kiss a frog and it becomes a prince, and perhaps more. And so I thought to myself, well, if I share 98% of my genes with Bertje, then we're a lot more than 98% different, actually. We've got remarkable similarities, but we're actually quite different. And at the time, I was learning about gene networks. So here I've got a little picture on my slide of a gene network. So every red dot here is a different gene, and every line is a locus of control, because there's a whole bunch of genes that are in fact we call transcription factors, for example. They're regulatory elements. They may turn something on or turn something off. And so one of the things that I learned is that what makes us different is not just the different genes that we have, which is what our DNA encodes, but also how they interact with one another. So there's a kind of a logic in here, just the same way that you can take a bunch of logic elements and turn it into a chip and make the chip be an oscillator or a switch or a whole bunch of different things. You can take the same units as proteins and these interactions. You can make it life do all kinds of different things that are really quite different from one another. In fact, think about this. Every cell in your body has exactly the same DNA. So the cell in my eye lens, has exactly the same DNA as the cells in my liver. Why are they different? Well, it's not the DNA that's different. It's the locus of control, is what things are turned on and what things are turned off. It's different between my eye and my liver. So you can have dramatic changes in the kind of output of these cells, depending on how what's turned on and what's turned off. And so a lot of the difference between ourselves and chimpanzees is not just in the kind of coding elements of the DNA, the genes that are turned on or off, or genes that are there, but also in what's turning things on and what's turning things off. So I got very interested in this question. But now if you look at this and you ask yourself, I have a whole bunch of little red dots, and I count how many different ways I can put little lines between them that tell me what some kind of regulatory control, what turns something on, what turns something off, then that grows factorially or exponentially. It just grows unbelievably quickly. And so the set of possibilities is very large, but that raises all kinds of interesting questions about how can you design these kinds of things, right? How can you, by random processes, which is what evolution effectively uses, stochastic processes that are used by evolution, how can you reach some kind of solution that really works? And so I thought, well, that's a really cool problem to work on. I have no idea how to attack it. So I'm going to try to find some simpler problems that are somewhat related and work on them. So that's what I started doing. Around that time, I moved from Cambridge to Oxford, and I'm working on these kind of questions ever since. But in the back of my mind has always been this question about what makes us different, and this question about kind of combinatorial explosions. So here's an example of something much simpler that I worked on initially, first started working on. So these are a related question. this is self-assembly. So how do things make themselves? So viruses, well, they're in the news, very big at the moment. So what is a virus? A virus is basically a piece of protein shell that encapsulates either RNA or DNA. And that little shell goes into your own cells, for example. It hijacks your cellular machinery, makes copies of the proteins, and then at some point kills the cell and pops out, and you get a whole bunch more, and then it multiplies in this kind of way. So what's fascinating about them is that they're often very simple, and they might use a whole bunch of copies of exactly the same protein to make their shells. Now, the shells, so that means, for example, let's say I've got 100 proteins in my cell. Maybe each virus is made of 12 different units, for example. And I've got 1,200 of these units, and they've got to spontaneously assemble into the right shape. So I got interested in this question of how these things self-assemble. Here's a little video of some viruses that I designed on the computer. Actually, my student Ian Johnson designed this as an undergraduate when he did this, actually. He designed this on the computer, a very brilliant undergraduate student who's now professor of mathematics. And he basically worked out a set of interactions between these little shells that meant that every time you start this program, even though the movement is entirely random, okay, you always get exactly these little TS12 forming in exactly the same way. And that's kind of cool, because if you think about it, the number of different ways you can put these things together is hyperastronomically large, and yet every time you randomly move them around, they form exactly the same shape. So this is a kind of principle that I got very interested in. And so I started working on, I thought I'm going to work on self-assembly. How do things make themselves? That's a super interesting question. And it's kind of interesting from a physics perspective, because there's a lot of statistical mechanics that you can apply to this question. It's a classic stat-mech problem. Another way of thinking about this is it's a little bit like taking Lego blocks. Okay, if I take Lego blocks and I put some glue on them, okay, and then I put them in my box and I shook the box, okay, what am I going to get? Well, if I just put a bunch of glue on them and shake them, I'm just going to get junk most of the time, because the vast majority of configurations are just junky configurations. But what nature does, and it does this all the time, is it puts these things together. They have a few different kinds of glues, because it's not that many, a few different kinds of interactions, that's it. It shakes it together, now it comes a really well-formed structure. Say, for example, like this train. So if I could figure out how to do this, if I could figure out how to put a bunch of Lego blocks together, shake it stochastically, and every time get this kind of train, that would be really cool. So no one, I think, can say that science is not, a lot of fun at the very least. So I got interested in this question. How does this work? How does self-assembly questions work? And I've been kind of running with this ever since. Now, there's one point I want to just remind you of, which I alluded to already, which is exponential growth is a lot faster than you might think. So here's a little picture from the Shamana, which is the Book of Kings by Ferdowsi. It's a great epic poem of the Persian people. And there's a picture in there of the players, the people in the Shah's environment playing chess. And the kind of story of chess, it's an apocryphal story, talks about a great Indian thinker who invents chess, and he goes to the king and he says, I've invented this great game called chess. And the king is extremely grateful for this great invention and says, well, I am a great king. I will reward you for whatever you want. Name your reward. And Sasa, the inventor of chess, says, well, great king, could you put one barleycorn on the first square, 2 on the second, 4 on the next one, and keep going this way? And at first the king is offended, because his great generosity hasn't really properly been acknowledged, until his accountants add it all up. And if you do that, if you take 1 barleycorn on the first, or 1 rice on the first one, and 2 in the second, you keep doubling on every single one of these squares, you'll see that it actually comes up to 18 quintillion of these barleycorns, which is many times the world's current annual grain production. So Sasa would have completely bankrupted the king had the king followed that on. And this tells you that exponential growth is much faster than you might think. So the space of possibilities that you typically have in a wide range of different problems, including biological problems, but also problems in computer science and problems in physics, is unimaginably vast. These numbers are just extremely, extremely big. And the chess is a very famous problem. Actually, along the Silk routes, there was a history of these. They used either 30 doublings or 64 doublings. These are typically the kind of interesting things. You can find it as far back as there's old Babylonian clay tablets from Mari on the border between Syria and Iraq from 2000 BC, where you really see the first 30mers. And this goes on. It's kind of fascinating little story. I don't know why they picked 30 and 64, but that's what they did. It was kind of a game showing the power of doubling. And the reason I point this out is because I got interested in the following problem, this biological problem, which is somewhat similar. So if you have RNA, I'll just remind you, because you may or may not remember this from your secondary school, but in RNA, you have 4 different letters you can have. And so if you have a strand of particular length, then there are 4 to the power L different possible strands that you could make. So that is kind of interesting, because if I just make a strand of length 55, for example, then I have 4 to 55 different strands. That would weigh 10 to the 10 kilos. If I made a length 79 strand, it would weigh more than the Earth. If I made a length 126, which is actually really short in RNA worlds, this would weigh more than the observable universe. Okay, and proteins, which are the main kind of molecular workhorses of the cell and in your body, they have actually 20 different letters they're made of. So any protein of length L can be made in 20 to the power L different types of proteins can be made. And they're just a length 37 peptide, which is extremely short, would weigh more than if you made every single peptide of length 37, it would weigh more than the Earth. If you made every one of length 58, which is extremely short, would weigh more than the visible universe. The mean length of proteins in our body is 476, according to Wikipedia. And so if you made every protein of that length, it would weigh 10 to the 500 times the mass of the observable universe. So I've gotten interested in this question. I call these hyper astronomical numbers, because you see I'm quickly reaching for metaphors that talk about the size of the universe. And so there's a kind of interesting question. These spaces are extremely big. These are 2 very important biomolecules, but their design spaces are unbelievably large. And so I'm thinking about things that self-assemble or things that do something. How on earth can I find a solution in spaces that are unsearchably big? That's the question that I'm really trying to get at. That's kind of the big question that I've been wondering about for now, ever since I kind of had my epiphany. And so, in fact, at Harvard, there's a really great scientist, Jack Szostak, in chemistry, who's a Nobel Prize winner, who's done a bunch of extremely interesting things. And he did an experiment back in 2000s that really caught my eye. So he and a colleague of his did a in vitro evolution experiment, so an experiment where you do evolution in the test tube, where they took a bunch of RNA sequences of roughly length 55 and basically tried to get them to do a particular catalytic activity. So it's a self-cleaving enzyme, but that's for physicists, that's not that interesting. For chemists, it's very interesting, but not for physicists. The point was, every time he did this, he would then what you do is you look for the sequences in your test tube that have the highest activity. You pick those, you mutate them again, and then you pick the best ones out of that, and you keep repeating that. It's like a little evolutionary process until you get a whole bunch of highly enriched sequences that have the activity that you want. And every time he did this, he found this particular structure right here. It's called the ham-red ribozyme. So this strand of DNA, which is a polymer, self-assembles into this quite well-defined shape called the ham-red ribozyme. And they published this and said that, how interesting, every time we run this, you get convergent evolution. So evolution converges to the same structure each time. And I read this and I thought to myself, well, that's surprising, because if you use length 55 RNA, then as I told you before, every single strand weighs 10 to 10 kilos. And I kind of worked out that, as far as I can tell, they used about 1/100 of a gram of material in total. So incredibly small fraction of that total space, and yet they found the same thing again and again. And that's kind of wild. It's a little bit like saying, you look for a needle in a haystack, and every time you look, you find the same needle, right? Now, if you found that, you think, well, something's up, because why are they finding the same structure, even though they've only searched an incredibly small fraction of the space? And so I got very obsessed by this problem and thought about it for a long time and gave it to lots of my students and made them work on it. And finally, we started making some progress on it. And the way we made progress, I wanted to explain a little bit about RNA. So here are the letters of RNA, 4 different letters, G, A, C, and U. And really, to first order, A and U bind together and G and C bind together, although G and U can bind a little bit as well. And so if I give you this sequence, this is a length 55 strand of RNA, then I can work out a structure, like for example, this hand-read ribosome structure, which is probably the free energy minimum of all the possible ways that this thing can fold on itself. And when it folds on itself, what I mean by that is that these things form bonds. Now, the sequence to 3D structure problem is extremely hard to solve. In fact, I would have said it until DeepMind's alpha, their very, very recent neural protein folding showed that perhaps it is solvable with neural networks. But this has been largely considered an unsolvable problem. Right? Even with the new DeepMind results, I would say that it's still very slow. But there's a simpler problem which you can solve with some basic methods of statistical physics, and that is, how does a sequence map to a simpler problem, which is what binds to what, or who binds to whom. So in this case, for example, this sequence you can work out relatively quickly, literally in fractions of a second, that this sequence will have this particular structure as its ground state. So it has 2 G's there at the end, as you can see. Then there's an A and a U bind to each other. The A's U's make little strands. Then there's a little loop here, and there's basically three of these little strands there of these little stacks, where you see, and 2 little loops, and 3 little loops. And this turns out to be the lowest free energy state of this particular system. And that's super. In fact, that's exactly what Shostak did, is he looked at his sequences, figured out what their secondary structures were, and worked out that each time the secondary structure was the one that corresponds to the hand-read ribosome. So then you could ask a question, well, if I just take a random sequence like this, okay, how likely am I to find this particular structure? Well, that turns out a problem that you can kind of solve. So I'll just want to walk you through how this works. So here I have a length 15 mer, so it's a very short little RNA, and this is what happens to be its ground state. Now you can kind of see, since I told you G and C like to bind to each other, I could flip the G and the C around, and probably this would still be the same ground state. I can flip this G and the C around, etc., and it would still be the same ground state. And so you see there's always some redundancy there. So multiple sequences will have this sequence as its ground state. And I can also represent this structure in what I call dot bracket notation. So if I start with a G, I say, well, this G is bound to something, so I'm going to give it an open bracket. This C is bound, this G is bound, this G is bound, so they each have open brackets. Oh, next 1, 2, 3, 4, 1, 2, 3, 4 are not bound, so there's 4 little dots, and then this one's bound again, so I'm going to put a closed bracket, and this tells me what binds to what, and I walk all the way to the end, and then there's 3 that are unbound. And so what I see is that this structure can be abstracted this way, and there are many sequences that give me this particular structure. And the question that kind of naturally emerges is, well, if I have a bunch of structures, do they all have the same number of sequences mapping to them, or is that mapping from sequences to structures very biased? And so we did this calculation exhaustively for length 15 RNA. So length 15 is 4 to the 15, which is 2 to the 30. So just nice thing back to the envelope, 2 to the 10 is 1,000, right? So 2 to the 30 is 1,000 cubes. It's 1 billion sequences. That's actually quite solvable on a computer. And what we found is that, this structure right here has 11 million sequences mapping to it, whereas this structure only has 2. That means there are only 2 sequences for which this is the ground state structure. And that's quite interesting, because it turns out that, first of all, we worked out that all billion sequences only have 431 structures. So then the sequence of the structure map is highly redundant. So even though the space is really big, the number of outputs that it does is relatively small. And just 26 of those take up 50% of the sequences. So 50% of the sequence that I take out of the billion, so 500 million of the sequences, map to this small subset of shapes. That's kind of cool because that tells me that I might be understanding something about what's happening in those search problems that Shostak worked on, and many others are worked on. And so we worked out a way of analytically doing this for much larger systems. So for length 55, you've got 10 to 33 sequences, which is unsolvable by kind of brute force computation, but you can do analytic calculations. And then we looked at, there's 504 natural RNA sequences that have been found in nature that we think are sequences that form structures. So just to take a step back, a lot of RNA in your body is messenger RNA. That means it takes a copy from your DNA and sends it to make proteins. And so that's its function. But there are also RNAs that are functional. That means RNAs that do something. And those RNAs will fold into these well-developed shapes. And so we looked at a database which has functional ones and took 504 different sequences for length 55. And what we found is here's what we call the neutral set size. So it's like the entropy of the sequence, which is the number of, it's the size of the number of sequences that map to that structure. So here, for example, this one has a neutral set size of 11 million. This one has a neutral set size of 2. Okay, and so here we have them, all of them, for all 10 to 13 different structures. And what we find is that the sequences that are found in nature are very highly biased towards this very tiny fraction of structures that have many sequences by mapping to them. It's as if nature's picking only these guys and ignoring these guys. And there's a very good reason for that, because when you take nature and randomly pick sequences by mutations, then you're overwhelmingly likely to get these structures and not these structures. Okay, and by the time you get to longer length, like length 55, which is already unsearchably large, you're only going to see these structures. So the only things that you're going to find are the things that are easy to find. Right? It's a kind of tautology, except that we've told you which things are easy to find. And that hemorrhoid ribozyme that Shostak found each time is one of those structures. So it's an interesting point. Why does nature keep finding the hemorrhoid ribozyme? In fact, you can find it in plants, in every kind of major animal clade. And the reason that you find it everywhere is because it's easy to find. And if you want a self-clearing enzyme, then that's what you do, apparently. Now, there may be, there's 10 to 13 different structures at length 55. There might be lots of other structures here that do the same work as this hemorrhoid ribozyme. Okay, so they can do the same thing. It's just that nature doesn't have time to find them. And so it's kind of fascinating. What this is telling us is that there's a small subset of structures that nature likes to use. Now, the difficulty a little bit with this dot bracket notation is it's very fine grained. And as the systems get larger and larger, you can kind of imagine that I might, shorten this or lengthen this little arm here without really changing the function very much, or shorten this loop without changing it very much, or the stack without changing it very much. And so there's a well-developed technique in the literature for taking a more complicated structure like this one, which is the length 100 structure, and simplifying it down to like a coarse grain structure, where you basically say, okay, what's actually happening here? Well, I've got a stack, and I've got a loop, stack, loop. And you can do this at different levels. It's a bit of a tedious but straightforward thing to do that a bunch of bioinformaticians have done. A lot of bioinformatics is actually quite tedious but important. So we can use it as physicists, but and very helpful. These guys, a German group has done this. And so what the nice thing about it is that a bunch of different sequences that might give me slightly different structures will all map to the same kind of coarse grained structure. That's also nice because that means if I look at natural structures, the structures that are found in nature, I'm more likely to find repeats of the same structure. So what we've very recently done is we've looked at these kinds of structures in nature. And what I have here on the y-axis is something extremely simple. So I just take random sequences of length 55, okay, and I fold them with this folding programs, and then I look at what the structure is with, in this case, level 3, which is one of the 3 levels, okay? And now I've worked out that there's probably 10 to the 7 different possible level 3 structures for this length 55 sequence. But when we looked in the natural database, there's only 28 are used out of the basically 10 million possibilities. And those 28 are the ones colored here in orange. The blue ones are basically how likely you are to get a structure by randomly picking sequences. So it's basically, these are the structures with large entropy, with many sequences mapping to them. And what you see is nature basically only uses this tiny subset of the 10 million possible structures. And this tiny subset it uses is the ones that are most likely to be found. And you can do the same for length 126. Remember, you might remember, if you have length 126 RNA, you made every possible sequence of that length. It would weigh more than the mass of the observable universe, so it's unsearchably big. And you have to course grain a little bit more. We think there's probably 10 to the 12 possible, so a trillion possible structure at this level 12 for length 126. But only 25 of those out of a trillion are used in nature. So this is kind of wild. Nature is only using this tiny, tiny, tiny subset of everything that's possible. Okay, it's amazing that it works with this tiny subset of all possible biological complexity. But this also explains why we keep seeing the same things in nature again and again, because nature is finding the same structures. I just want to point something out to you is that these analyses I've done have completely ignored natural selection, which is obviously a very important part of evolutionary theory. And I'm just pointing out something different, which is you can only select for function on those things that actually pop up in the first place. So in evolution, you've got really two steps. One is you have random mutations that give you some kind of variation, so some kind of difference in an organism or in a phenotype. And natural selection can enact on what is given. And what I'm telling you is that in this system, at least, there's an extremely strong bias towards a very small subset of structures that are really easy to find. And those are the ones that natural selection then works with, and that gives us the complexity that we see around us. This is super interesting for questions like the origin of life, where did life originate from? Because it tells us that there's a small subset of things that you're going to find again and again. Now, this raises a question.
Professor Louis.
Why is this a strong bias? And this is a typical physicist question. So it's all fine and well that I showed you this is true for RNA.
Sorry, Professor Louis.
Yes, go ahead.
Sorry, we have a question from Mikia if you want to ask. From Mikia,
Yeah, go ahead. Yeah.
It was interesting to see the analysis you did a few slides back.
Yeah.
My parents are my dad's a bioinformatician, and so like they deal a lot with proteins, protein sequences, and so I have a little bit of like bleed over knowledge. I was wondering if you did any analysis on the probability that a single nucleotide polymorphism would affect the phenotype of a given strand of RNA.
Yeah, so typically these RNA strands are very robust. That is to say, most single mutations don't do anything to them. And that's kind of a fascinating property that they have. So they have what's called, there's a lot of neutral evolution. That means that you have substitutions that don't affect the outcome. And this is known, across in molecular evolution, is very well known that you have a very large number of neutral evolutions. And your parents, if they do bioinformatics, almost certainly use this to do, for example, evolutionary inference. So to look at what's related to what, you can look at neutral evolutions. Okay, is there any more questions? I see, and how do we know we've discovered significant numbers of these structures used in nature? Well, so what we did is there are bioinformaticians that have worked out what they think are functional RNAs. And so what we simply did is we looked at all these functional RNAs and asked ourselves which structures have they found? And we found that those functional RNAs only pick a very small subset of structures. And the interesting thing is that we can predict exactly which structures they're going to find by just randomly sampling sequences. So just ignoring the biology entirely. That's a very kind of physicist way of approaching it. But it tells us that nature is only using a very small subset of all the complexity that's available to it. That's what we're basically saying. Hope that helps.
We have another question from Yash. It seems like.
Yeah, go ahead.
So in the previous slide, you talked about there's some of the crosses were not circled with higher probability. Do we
Yeah.
know why?
No, no. So the point is, there's a few of the predictions that we make that are not seen in nature. And we're not really sure why. Maybe if you keep looking longer, you'll, at more organisms, you'll find them. Maybe there's some other reason for it. The main point we're just pointing out is that out of the 10 million length 55 or a trillion length 126 structures, we can the ones that are found in nature are a tiny subset of those, and we can tell you with a pretty high probability which ones are going to be. And that's completely wild and crazy, I think. And so what that is telling us is that the mapping, okay, from the space of sequences to the space of structures is extremely biased to a very small subset of structures. And that raises a really interesting and important question, I think, which is why on earth, or why is this a strong bias? And is this, something particular to RNA, or is this a property of the world? I wouldn't be asking you this question if I didn't think it was a property of the world. And so I'm going to take a different tack and talk about something completely different for a second, and at the end of it, hopefully you'll see what the connection is. So,
sorry, a really quick question before you continue. I'm just curious. Obviously, from a physics perspective, there's this idea of symmetry breaking. Do you think that, first of all, is self-assembly a form? Can it be considered a form of symmetry breaking? And adding on to that, do you think it would be possible for, I guess that's a moot point. I guess my question is, does the chirality of the universe, right, with respect to the handedness of, like DNA and all this stuff, affect the way in which, we have certain biases towards self-assembly?
Yeah, that's a good question. So the bias towards certain symmetry of chiralities is a kind of symmetry breaking. And I don't actually know why that has happened. I think it's a complicated question, which I don't have an answer. I don't think you can think of self-assembly in itself as a symmetry breaking, although you can have symmetry breaking in self-assembly, if that's helpful. So you can have self-assembly to different kind of structures. One of the confusing things is if you just do random self-assembly of many biomolecules, then you'll have equal chiralities of left and right. Whereas in nature, you just see one. And the question is, why is that broken? And the answer is, I don't know. It's a super interesting question. Another deep question that's well worth spending a lifetime thinking about.
So we'll take one more question from Mark, and then after that, we'll let Professor this topic.
Like, should I unmute and ask the question that I put in the chat?
Yeah, but probably unmute. That's probably easier.
Yeah. Okay. So I mean, you mentioned you seem to have specified functional RNA for some reason, and it leads me to think that you are specifying that because non-functional RNA sequences are not heavily biased towards, the very likely sequences of structures. The structures formed by non-functional RNA. Does this question even make sense? Hang on a minute.
That makes sense. That's an excellent question. So what I'm saying, I'm picking the functional RNA because those are the ones for which we think that the structure is important. For the non-functional RNAs, for coding RNAs, as far as we know, the structure is not that important. It's still the case that if you take non-functional RNAs and you look at what structures they make, they're going to make a very small subset of structures. But typically, they're not studied because they're not thought to be important.
But I mean, if the way you're getting which structures are found is purely by statistical methods, wouldn't you expect that non-functional RNAs are like fairly random and like they should follow the same thing? Is there any reason that
they do? They probably do follow the same thing, but as far as we know, that their structure is not that important. They just get sucked up by the ribozyme, right, and then decoded, and probably the structure gets destroyed at that point. But that's the first order answer. The second order answer is there's all kinds of ways the structure matters because of alternative splicing and introns, etc. But the first order, what the idea is that functional RNA, which can be structural or it can be catalytic, that there we think the structure really matters, which is why we care about the structure there, which is why typically that's what's studied.
Okay, I'll stop. Yeah. Thanks.
Great. So the question this kind of raises is, why on earth do you have this kind of super strong bias, right? Why is this the case? And so I'm going to talk about this, and I'll probably finish with this story because I think I'm getting close to the end of my time. So here's a question that you've probably thought about at some point, which is how likely is a monkey typing on a keyboard or some very large ensemble of monkeys to type out a particular sequence. So let's say that I've got monkeys typing onto a word processor, and how likely to type the function pi. Well, if it's an n keys on the typewriter, so say 50, and I want to get the first m digits of pi, then it's 1 over m to the power m plus 1, because I got 3.1, right? 3.14. So for 3.14, I need to get 4 things right. And so the chance of getting pi right is extremely small for longer versions of pi, but it's actually true for any sequence of that length. They're all equally likely or unlikely. That's the famous trope of monkeys typing on a keyboard. But I can also ask a different question. What if a monkey types into something like C, right, a computer program? Well, then something very different happens. The monkey might accidentally type a program which will generate pi. So there's a competition, in fact, to write the shortest C program that will generate pi up to a certain number of digits. And here is a 133 character code that will generate the first 15,000 digits of pi. Use something called a spigot algorithm. And so the monkey might accidentally type these 133 characters completely randomly, and the first 50,000 digits of pi will spew out of the computer. So if you're looking at the output of these monkeys typing on keyboards, you'll be able to tell whether they're typing onto a word processor, and so that every sequence of the same length equal likely, or whether they're typing into something which is generating outputs, because you might see certain computable numbers, certain kind of algorithmic numbers like pi will appear with much higher probability. Or think of 0, 1, 0, 1, 0. Anything with patterns are going to appear with higher probability, because you might accidentally type that out. You have to wait in practice for a very long time, but let's assume you've got an extremely large number of monkeys, and you can wait many times the length of the universe. And this idea has actually been beautifully formalized in a branch of mathematics called algorithmic information theory. So you're probably used to kind of Shannon information theory, which is kind of theories about information of distributions. And what Kolmogorov and Chaitin and a few other people were thinking about back in the 60s was, how do I find the information content of a single sequence? And intuition, it goes a little bit like this. If I see a sequence like this, 0, 1, 0, 1, 0, 1, 0, 1, 0, and just repeats it, then I can compress that sequence by just writing print 0, 1 50 times, for example, and I'll get this particular sequence. But the sequence below it, as far as I can tell, as I know, has no shorter code and simply prints the sequence. And so what Kolmogorov and Chaitin and others showed is that this is a very deep concept, that there is a beautiful idea that you can take a universal Turing machine, which is a computer that can do anything, and look for the shortest code that describes a sequence, and that will be called the Kolmogorov or the Kolmogorov-Chaitin complexity of that sequence. Now, there's really deep links to foundations of mathematics, so you can prove, in fact, that you can never calculate the Kolmogorov complexity in an algorithmic way, because you never know whether there might not be a shorter program that you haven't found. This is actually linked to Gödel's undecidability problem, and this is very beautiful and interesting. But you can talk about this thing, just like you can talk about infinity, although you can't calculate infinity. You can still talk about an object that you can't necessarily compute. And it gives lots of interesting intuitions, like what's the definition of a truly random number? Well, a random number is a number for which the shortest code is just print the number. Okay, so its Kolmogorov complexity is always at least the length of the number, because you've got to print the number. Other interesting intuitions are like this: the complexity of a set can be a lot less than the complexity of its elements. So if I say to you, give me all the numbers from 1 to googolplex, that's a very short description. So its information of that set is low. But there are individual numbers in that set that may be very complicated to describe, right? So sets can be much simpler than their elements. And so there's all kinds of funny things that throws up. Now, why am I talking about this? Is actually the idea of Kolmogorov complexity was made by an American iconoclast, Ray Solomon, who actually lived in Boston and kind of was an independent thinker there, who, well before Kolmogorov, was asked the question, what if I have a universal Turing machine and I just randomly type codes into it? So I'm going to simplify my life. I'm going to make my codes be binary codes. I'm going to type into this program, and I'm going to ask myself, how likely am I to get some output X, like pi, if I type on a universal Turing machine called U? Well, I'll randomly type all codes, and every code that gives me pi, I'll add to my probability. And since my codes are binary, they've got 1/2 to the power length L is the probability I get the code. So if the code is 0101, it's 1/2 to the power 4, etc. So, well, he said, that's interesting. What's the most likely output? Well, the most likely output is going to be the shortest code, right? And that's 1/2 to the power Kolmogorov complexity. So in fact, he had the idea of Kolmogorov complexity well before Kolmogorov. And it's called Kolmogorov complexity because Kolmogorov was a very famous mathematician and Solomonov was not famous. This is the Matthew principle. To he who has, more will be given, and to he who has not, even what he has will be taken away. From St. Matthew. But you know, it's a beautiful idea. And I'm pointing this out because this is starting to look a little bit like these monkeys on keyboards, right? So you're randomly typing things in, and what are you going to get? Most likely, you're going to get things with high Kolmogorov complexity. And this idea that Solomonov came up with, Marvin Minsky, is a great father of modern AI, who was just across the river from you at MIT, said not long before he died. It seems to me that the most important discovery since Gödel was discovery by Chaitin, Solomonov, and Kolmogorov of the concept called algorithmic probability. Everybody should learn all about it and spend the rest of their lives working on it. And so it turns out that this actually is interesting because it's a big Boston story. So, Levin, who's also very famous for P. Resenpi, who also lives in Boston at Boston University, worked out that not only was this a lower bound, which is what Solomonov found, but there's also an upper bound, which is by this 2 to the minus k. And this tells us very interesting. This tells us that complex things are much more likely to appear than simple things if you randomly type them in. So by complex, we mean things that have long descriptions, and by simple, we mean things that have short descriptions. And I think this coding theorem, which I've just written down in this really simple way, should be taught to undergraduates everywhere in the world because it's a really deep and powerful thing. The problem, of course, for applying it to something like RNA or other evolutionary systems is that many systems are not universal Turing machines. Okay, that's like a computing where it has basically infinite power. It can be anything computable. I told you that Kolmogorov complexity was formerly uncomputable. This is one of the reasons why it's kind of stayed in abstract areas of mathematics and computer science for a long time, because there's interesting links to foundations of mathematics, and it only holds technically in the limits of very long strings. So I started thinking, I've been thinking about this for a long time, and I finally found some students to help me with this. I'll skip the proof sketch, but we basically proved that we can't prove a lower bound, but we can prove an upper bound that tells you that even for non-universal Turing machines, where you make an approximation to Kolmogorov complexity, you can still write this scaling argument down, that the probability I get a certain output X scales like 1/2 to the power of the Kolmogorov complexity of that object. It's a very typical physicist type of an approach. Okay, so we see a scaling argument which we can prove in a certain limit, and we say, let's try using it outside the limit where we can formally prove it in the way a mathematician would, but we can still prove it heuristically, and let's try and use it. And what we find, if I look at the RNA, is something really kind of exciting. So here, I show you different structures of RNA. This is obviously a very simple one, nothing binds. This is a very complicated one with lots of different ones. And then I've got on the y-axis here, I've got the complexity of these, which I do by just compressing them. So this is very compressible. This is not very compressible. It's like a proxy to Kolmogorov complexity. So what's the shortest code I can write to describe the sequence? And what I find is that sequences that are short descriptions are very likely to appear, and sequences with complex descriptions are very unlikely to appear. Okay, and that's exactly what Solomonov and Levin had already predicted. We've just applied this with some twists to the RNA. So why is the RNA system so incredibly strongly biased for a small subset of structures? Well, because this bound is is on a log scale, right? So they're exponentially, every time you add one bit of information to describe this system, your probability goes down by a factor of 2. And so very rapidly the probability of getting certain things drops. And this principle works for a wide range of different systems. So I showed you RNA. So this structure is complex. It's unlikely to appear. This one's simple. It's more likely to appear. I can do it for a wide range of systems. So here is a set of differential equations. Here is something called L systems, which are used for computer graphics to make all kinds of shapes. If you watch the movie Avatar, you'll see these beautiful forests which are made with L systems. If you randomly pick L systems, you'll find that simple L systems are exponentially more likely to appear than complicated ones. So that's basically the story that we found here. And then I've got a bunch of other examples, which I think I've run out of time, which is fine because the point of this exercise is not to give you an overview of my work, but just to give you a story that's linked to my own life. And I'm very pleased with this thing. I think it's just now coming to fruition after working on this for a very long time. And I was interested in this question of where does biological complexity come from? I then realized that the space of biological possibilities was so big that it was unsearchably large. And yet in experiments like Josh Shostak's, we find the same thing appearing again and again. It was a great conundrum. I thought, well, how on earth could that be? I thought, that's what I'm going to work on. It was a crazy problem, an unfashionable problem. It still is an unfashionable problem because people don't think it's weird. But I think it's super important. And so I spent a lot of time thinking about it, and finally actually had a very bright mathematics student who put me onto the coding theorem and onto algorithmic information theory. I realized that I had to work on that for a long time to make it palatable to my physics sensibilities. And then when I tried it, it seems to explain this particular property. And in fact, it turns out it explains a whole bunch of other self-assembling systems. So self-assembling protein structures. In fact, if I go all the way back to the beginning, I told you about that gene regulatory network with all the different interactions, which has the same combinatorial explosions of possibilities. These ideas from algorithmic information theory, we believe, explain why you see the kinds of structures of gene regulatory networks that you do in nature. So I'll skip the rest of the story. It turns out this also applies to deep learning, so to why neural networks work well. We think we can explain why neural networks generalize so well using these same principles. And the last thing I want to say to you, and this is a really important point, is I've talked about this as if I did this, all right? Very often I use the we, it sounds like the royal we, but here's a subset of the people that worked on this problem over the years. So Ian was the first student that worked on this. He did a virus simulation, all the way to Vaibhav, who's graduated from Harvard not too long ago, and has been working with me on these problems and doing some really brilliant work. And all these different students over time have worked on different aspects of it. And one of the great things about being in academia, which is why I sometimes pinch myself that I'm actually being paid money to do this, is that you get to work with really, really smart young people, often who are unencumbered by, tradition and prejudices, and so will try completely new, crazy things. And that's how you really make progress. And so one of the great things for you, if you guys go on in research, I hope you will, is you'll become part of one of these groups and work with other people. And you see that knowledge is generated not by individuals thinking about this stuff by themselves in a dark room, but by groups of communities working together and thinking about stuff together. So with that, I want to thank you very much for your time. And I hope that I entertained you at least with a story that I'm very passionate about and I'm very excited about. It's not traditional physics, but actually it has a lot of physics in it. So thank you very much for your attention. I'm happy to take some more questions.
Yeah, thank you so much for joining us. And before we go to questions, I just want to make sure before too many people leave, I want to thank everyone for joining us today, especially if this was your first time. We're so glad you're here. To the folks who were publicizing this event at their respective schools, thank you so much. We hope you'll continue to do so. And also feel free to invite as many people as you can. If you have friends, if you have colleagues, if you have folks at contacts at other schools, you really want as many people here as possible. But just thanks everyone. And a big thank you to you, Professor Louis, for this really just engaging and enjoyable talk.
So I took a bunch of questions already, but I'm happy to take a lot more. So go ahead.
If people have questions, feel free to raise your hand or throw them in the chat.
I have one question actually. are there any discrepancies between the like algorithmic and entropic definitions of like information content? Do they clash?
Yeah, that's a super interesting question. So they're two very different things in the following sense that, entropy is really a description of some kind of distribution, right? So it's a measure of surprise. So for example, if I have a distribution where I have a whole bunch of copies of the same thing, let's say I have a distribution of books, okay? So I've got a lot of multiple copies of Ashcroft and Merman and a few copies of Kittel and a few copies of some other books, then I can write out an entropy of that, which tells me how frequently how likely I am to get each of those different books. And if you say, give me a book, how likely it is to be Ashcroft and Merman versus Kittel or some other book. But that same distribution works for apples and oranges and lemons. It doesn't really matter what the thing is made of. The entropy describes just how frequently something appears. So algorithmic information is interested in the individual things there. It's interested in what's the information of the actual object. So a book will be much more complex than, say, a much simpler. A long book will be more complex than a short book, etc. And so they're kind of two very different things, except that if I'm asking you, for example, what's the algorithmic information of a single object, and I can say write down it as a binary sequence. If that binary sequence has low entropy, so that's, for example, all zeros, then it probably has a very short description, which is print 0 100 times, or however long my sequence is. And so there's where the link with entropy comes in. So entropy gives me a kind of bounds on it. But there can be structures that actually have high entropy, but nevertheless have a simple description. And that's where AIT is different from entropy. But the link between the two is super interesting. Even though they're kind of describing different things, there's a lot of beautiful similarities back and forth between them.
Great, thank you so much.
People don't have questions. I might have a few questions.
Right.
But wait. All right, cool. So earlier on, you had the simulation of the kind of virus self-assembling.
Yeah.
You mentioned that there's some sort of interaction rules that you can design, so that you can guarantee some sort of convergence to like the same. You said the same kind of end configuration.
Yeah.
How do you like what do these rules look like, or like how can we make?
Yeah. So there's lots of different ways. That's a really good question. Thanks for asking. So the rules, typically interaction rules. Like let's say I've got a bunch of different particles. I'll say, can A bind to B, B can bind to C, right? And I can make up lots of different ways of making those kinds of rules. Maybe I'll just show you very briefly a picture. It's probably quicker. It'll be quicker for me just to share my screen very briefly. Again, in fact, I'm still sharing my screen, aren't I? Good. So I'll just go back to, I skipped over this, but I'll just give you one example of this really quickly. So here's a system I played with a lot. It's called Polyomino. So these are little squares, okay? And they have sides on them. Do you see that? Okay. I can set up a set of rules that tell me what binds to what. So if I set up always this one first and then let these ones randomly join, and I got this set of interactions, so A binds to C here, okay? If I put one down, then only one that's going to bind to it are the threes. The threes are going to bind because they've got the only one that has C's. They've got D's on the sides. 2's got a B on it. B binds to D, right? So the 2's will go there, etc. So with this set of particles and this set of rules, I'm always going to assemble into this structure. So that's an example of a self-assembly rule that tells you that these particles will always self-assemble into this structure. And your body is full of self-assembling structures that basically do this. So proteins have little side interactions, so they're sticky sides that stick in certain ways that will give you the same structure. And you need to get exactly the same structure each time.
So is this sort of like in like the physical world, like this sort of like rule would be something like some sort of bond is more favorable?
Yeah, exactly. So the rule would be, for example, this is a protein that has sides on it. It has a little patch that binds to, so in this case, the A, right, binds to C, so that the A patch binds to the C patch. So in the case of protein, that would be some amino acid combinations. But you could do this on a large scale. You could make, structural things that you could make you could literally make physical squares with little sides on them with different kinds of glue. Okay. And that would make that. So here's the interesting thing about this. I can turn this question around, okay, and say, if I give you this structure, okay, can you find the rules? Okay. And then there's a kind of cool thing, which is that if I do that and I just randomly search in the space of rules, that I'm going to find that structures that have nice symmetries, that have short Kolmogorov descriptions, are very easy to find rules for. And structures that are complicated, that have large Kolmogorov descriptions, so it takes me a lot of Kolmogorov information to describe the structure, have very long rules. And so that's kind of a story.
So, but you know the rules have something to do with like the sides, or like what
the rules are to do the sides, but you don't know, let's say, what this matrix is or how many blocks you need. So if I give you the structure, I can ask you, so I can always make this structure, okay, by making every single one of these squares different from the others, right? So I can make a very large number of squares and a very large number of sides, okay, so that each one is different. It's like an addressable self-assembly. Every little thing has its own little address in space. But that's not kind of boring, right? I'd like to make it with a small set of rules. And for this particular structure, this set of rules is the shortest, the smallest set of rules you can make that will make it. That's what we think, okay? Can't prove it formally, but we're pretty sure this is true.
Okay.
And that's like the Kolmogorov complexity of that structure, is the shortest set of rules that will make it, okay? So this is a relatively simple structure because it's got a relatively short set of rules. Ergo, if I randomly mutate these sides, I'm likely to get a shape like this, but I'm unlikely to get a shape that's complicated.
The other question I have has something to do with like kind of like landscapes that you didn't really, maybe you didn't really touch about on this, but so I think the surprising part of what you were saying is that like somehow this is a very large space. So even though there's like a well-defined ground state, finding the ground state through like some sort of non-equilibrium dynamics is hard to find, right?
Yeah, kind of... no, that's good, super. So you've touched on a very important question that I've completely missed out, which is that, you know, in real life, you're not randomly searching through the space of these things. You're starting somewhere, okay? So you've got a particular RNA structure, and then you're slowly mutating through some kind of fitness landscape, right? And the fitness landscape basically gives you higher or lower fitness depending on what mutations you have. And what we've done a lot of work on, but I've not talked about today, is can you predict what happens on these complicated fitness landscapes? And the argument that we're making is that this entropic, so when you think about in evolution, people talk about fitness all the time. Biologists talk about fitness all the time. And that's like energy, okay, in statistical mechanics. Fitness is like the energy. It's like how likely, how fit is something, like how was the energy of the state. But we physicists know that entropy plays a really important role. It's like, so I can have a high energy state, which is very rare, and a low energy state, low energy state, which is very rare, and a high energy state, which has many ways of being made. And my system may actually be in the high energy state because its entropy is larger. And so our argument here is something like this. Simple structures have a large number of rules mapping to them, a large number of sequences mapping to them. They have a high entropy. So even if their fitness may not be the highest fitness, they're much more likely to appear and to fix in nature. And this is super helpful because the fitness landscape is like the energy parts of your story, whereas the neutral sets, the number of rules mapping to something, is like the entropy of the story.
Couldn't you look at something like a free energy or something like that?
Exactly. So what you need to look at is some kind of free energy, and what we're pointing out here is that because of the exponential nature of the difference in size of the number of rules that map to a structure, these are systems where we claim the entropy often dominates over the energy. So although what you need to write down is a free energy, or a free fitness would be the way of talking about it, these systems are often working in the regime where the entropy dominates.
So is the picture like there's some sort of funnel that leads to the ground state, kind of like there's this like Leventhal, whatever.
Yeah, exactly. So okay, so you're obviously a bit further along than the average undergraduate, but yeah. So when you think about this in terms of self-assembly of an individual structure, like how does a protein find its ground states? And Leventhal famously made a kind of hyperastronomical numbers argument where he pointed out that the number of possible configurations of protein is incredibly large. How does it find something? There must be something about its search down to the bottom. And that's true for this kind of self-assembly systems. What I'm looking in design space. I'm asking about designing things. And there, it's not the same story. It's in fact a simpler story, which is simply that the change in the size of the basins vary by so many orders of magnitude that you're just going to fall into the big basins. And although there are many, many, many small basins, you're never going to find them. Whereas the Leventhal story is really also a kinetic story, right? So what you need to design a good folding system is not just that particular ground state is the actual ground state, but that you can actually find it.
Can I ask a follow-up question about that?
Yeah, go ahead.
So you talked about it earlier, just that like these more likely bonds, I guess, the ones that require less energy are going to be the most likely. What was your process, I guess, for finding a translation between this more physical and like way to actually find? And I guess obviously it's tricky, but you have a way to quantize what requires less energy than something else. How did you translate that to a lower K of X value, a lower probability? Because you
so maybe just see if I understood your question. So I think what we're saying here is something kind of, so I've illustrated this with the example of the RNA for you. And what we're saying is, look, I can take an RNA structure. I can ask myself how much information I need to describe that structure. And if the information to describe that structure is low, then I'm going to predict for you that the number of sequences mapping to that structure is going to be very large. That means the number of sequences for which that structure is the ground state is very large. And that is basically just from this very, very high level Kolmogorov coding theory or argument. And so what I didn't say, which I think I should have said, is that what is happening in these RNA systems? What's the code? What's the monkey typing? The monkey typing is the sequences. What is the computer language it's written in? Well, there's a language there, which is a small subset of rules that are the free energy rules that tell you what binds to what and what those rules are. That set of rules is a relatively short little computer program that you're typing codes into. That program takes that sequence and then spits out a free energy minimum. Just the program is, here are the rules for what the free energies are for different ways of binding, and find the lowest free energy. So that's like a very simple computer program. And so what this Kolmogorov, coding theorem tells you is that if you have a simple programming language and you throw a bunch of programs at it, then you're much more likely to find outputs that have simple descriptions.
So I kind of missed at the beginning. I was just kind of struggling to understand what was giving the mass in your metaphors about like a protein of a certain length, and you said that its mass would be like all of the observable universe. So what is giving that mass?
Yeah, that's a good question. So the mass is, so if I have a length 53 protein, okay, and I make every sequence of that length, so I've got 20 different amino acids. So at each point I've got a choice of 20 different amino acids, okay, so 20 times 20 times 20 times 20 times 20 times 20. So I can make them all with glycines, which is one, or all, and I can start, if I make every single one, then it weighs more than the mass of the length 58. So right, make every length 58 will weigh more than the mass of the observable universe. And that's because this is a little bit like the exponentiating problem. The exponentiation grows much faster than your intuition might normally be used to. Right, so even extremely short, 37 amino acids in a row, there's 20 different amino acids. If I do every possible combination of amino acids of length 37, it weighs more than the Earth. That's kind of wild, right? Maybe you're better than me at exponentiation, but I was surprised by how short those sequences were. And so the point is, if you run an evolution experiment many times, like Josh Shostak does, and you keep finding the same structure coming out, normally you would say, well, it's because you've searched through the whole space and you keep finding the same thing because it's the best. But I'm just telling you, there's no way of searching those spaces. Those spaces are so big, they're unsearchable. And yet you find the same thing again and again. Well, that's very odd. Well, the only other reason can be this, that actually searching the space is highly biased. You keep finding a certain small subset of things again and again and again. And so we found that empirically in the RNA, and then we have a very high level explanation of that, which we use this algorithmic information theory to describe. And the high level explanation is that things that are simple to describe are much more likely to appear on random inputs.
Thank you. I have another question, but I don't want to hog if anyone else has a question. Okay, I guess I'll just go for it then. So so you're talking about it's interesting that you started talking about these hyper astronomical numbers and then said it's amazing that only a small amount of these are found in nature, but you could have started kind of the other way and said, like, will we ever, because I guess that my question now is about the database of functional RNA, and if it has such a small amount in this massive space of possible combinations, and the ones that are actually listed in the database are only a small set of these low energy states, why are you concerned then with, or not why are you concerned with, but it just seems interesting to go from such a huge space, and it's amazing that we have these small amounts, but if we had the functional database, or if we had the database of functional RNA first, then it's not really surprising, I guess, that we only observe these low energy state RNAs.
Yeah, so what I would say is, if you take the length 126, for example, I would argue that there are about 1 trillion possible short length ground state systems that you could find. Okay, so if I only have 1,000 of them in my database, yeah, I'm going to get, if there's 1 trillion possibilities and I only have 1,000 examples, I'm going to find each of them is going to be different. Right? It would be super surprising if you took 1 trillion, out of 1 trillion possibilities, you took 1,000 samples, and I told you exactly what those 1,000 samples were. You would think I was tricking the system. Right? Mm-hmm. That's what I'm saying. So I'm not saying it's surprising there's a small number of them in the database. I'm saying it's surprising that I can tell you which ones you're going to find.
Yeah. Okay, cool, great. Thank you. Super interesting stuff.
I have a quick question.
Right.
First of all, thank you for the talk. It was really great. Second of all, I've heard of life kind of being described as going against entropy. We're trying to go into a very low entropy state, like a human would not just randomly appear out of nowhere, right?
Yeah.
So then I find it very surprising that on the very small scale, on the RNA scale, that we find these structures arranged according to their entropy. And I was wondering if you've thought about this, found a way to reconcile the very going with entropy nature of the small scale with the very going against entropy nature of the larger scale.
Yeah, so I think that's a really interesting question. So I have thought about it, and I don't have an answer to the question in a meta way. I can say this, which is that what I'm saying is that the structures that you're likely to find are structures that are simple, right? So they're structures that have low description. Those are like low entropy structures, actually. And so one of the things about us is we're actually a low entropy state, right? We're a unlikely state. But the other thing which is interesting is that these kind of structures, that these structures, so far I've only talked about molecular structures. So what's nice about them is because they have many sequences which map to the same structure, that means they're very robust. If I have a random mutation, I'm likely to get another sequence that gives me the same structure again. And that's nice, because if I want to make something more complicated, what nature often does is it takes solution A and solution B and then binds them together. And what you want to do if you've got solution A and solution B and bind them together, you want to change both of them so they can bind together without changing A or without changing B. Right? So robustness is a very useful quantity to allow modularity to arise. Because if you're robust, then you can change your things a little bit without fundamentally changing what you do. So you change A and B, both remain A and B, but if you bind them together, then they become this larger modular whole. So one of the things about us, our biology, is that our biology is very modular. In other words, we've got lots of modules that act independently of each other and that are connected together, but still work together. And that needs a certain amount of robustness. So that's my very, very hand-waving way of trying to say, why does this bias towards simplicity give us the biological complexity that we see, which I think is what your question is asking.
Yeah, that was the essence of it. Very interesting. Thank you.
Oh, thanks.
Okay, I think we'll shut off the recording. Also, it's getting kind of late in the UK, I imagine. So,
Yeah.
but if you want to stay here, I mean, there's no.
I probably will head up. My kids are going to get up very early tomorrow morning, so I will have to